Hey guys! Today we're going to talk about topic 3.4, inheritance. And the essential idea for this particular topic is the inheritance of genes follows patterns. So, the genes that you get from your parents are determined through a couple of different ways. We talked about that in meiosis last time. But how those genes combine and how they are expressed can follow a pattern. So as we get started, I just want you to know that if you follow the understandings in the handout, you'll notice in the handout they don't necessarily all go in order. On the handout, they are listed in the order that we're going to talk about them in today's lecture. Okay? So we're going to start by talking about inheritance and alleles. And if you remember, an allele is a version of a gene. Understanding number 1. Mendel discovered the principles of inheritance with experiments in which large numbers of pea plants were crossed. Gregor Mendel, who was born in 1822, was an Austrian monk known as the father of genetics. Now, this is important because remember, 1822 to 1884. So he was conducting these experiments from around the 40s, 50s, and 60s. That's 1840s, 50s, and 60s, maybe a little bit later. We knew nothing about DNA. We knew nothing about nucleotides. We knew nothing about that kind of stuff. And he still managed to develop the two major rules that we use today when we're talking about inheritance. And he did it strictly through experimentation. He crossbred plants and looked for those phenotypic ratios that we talked about in our last lecture. So, as I said, he was a monk and he did a lot of work in the garden. And he noticed that pea plants in the garden had characteristics that were sometimes passed on to their offspring, but not all the time. So he was interested in figuring out how often does that occur and what are the rules behind that inheritance. So to study how Professor Pea Pants, I've been reading too much Captain Underpants lately. Sorry. I do have a 9-year-old. Okay, so to study how pea plants pass on traits, he bred plants that were pure for 7 different traits. Now, when we say pure, we mean homogeneous. But remember, he doesn't know what genes are and he doesn't know what alleles are. So when he means pure, he means he has a plant and it has a particular trait and he's bred it 1,000,000 times and he is convinced that the parent plant has only that trait to give. Okay? So when we know pure means homogeneous, meaning 2 alleles of the same type. And we'll talk more about that a little bit later. You do need to know these 7 traits. I have seen essay questions on the skills test that ask for, outline how Gregor Mendel experimented on pea plants and determined the law of segregation. So here are the 7 traits. We have seed shape, which is round or wrinkled. We have seed color, which is yellow or green. We have flower color, which is purple or white. We have pod shape, which is inflated like a balloon or round, right? You can see that down here. Here's our inflated one, and here's our constricted one. Did I say inflated or round? I'm sorry. Inflated like a balloon, all puffy, or constricted, like you can see all the different peas in the pod, right? That's constricted. And then pod color, yellow or green. The 2 other traits deal with not so much the seeds or the flowers themselves, but the flower position. We talked about this last time. If the flower is midway up the stem or along the stem, it's axial. If the flower is at the end of the stem, it's terminal, like the end of a subway or a bus line. You get off at the terminal, right, or the airport. And then there's stem height. Is it a tall plant or is it a short plant? Okay, those are the 7 traits you need to know. So Mendel crossed huge numbers of plants. Now, this is replication. You always need replication in science, right? You don't want your data to be a fluke. And when we say large numbers, we mean thousands upon thousands. And he crossed plants with different characteristics to determine which traits would appear in the offspring. And this is how he got those phenotypic ratios that we talked about last time. That 9:3:3:1 when you're crossing dihybrids, that's a phenotypic ratio. He looked at the traits he could see in the parents in the first generation, and then he would cross that first generation. It's not weird, it's plants. And he would look at the phenotype of the second generation. Okay, so before we go too much further, I want to show you a little bit of notation, and I'm practicing using a new pen, so it might take me a minute. Whenever we're doing genetics like this, right, our parent generation is going to be denoted by a P, right? And then you can see here, F1, that's the first generation, F1. And then we have F2, right? F2. So we have 3 generations. P for our parent generation, right? F for our first generation, and F2 for our third generation, okay? So his observations after doing these experiments crossing multiple generations of plants allowed him to discover the principles of inheritance, which are discussed in this topic. Now, the IB curriculum doesn't do a great job of talking about the names of these particular principles, but we did talk about them last week in our notes. So really what we're talking about is the law of segregation and the law of independent assortment. These are the rules by which traits are inherited. So you can kind of think of genetics as like game theory, right? And everything is a rule of a probability, and these are kind of the board rules. These dictate how the probabilities will fall for certain things happening. Okay, understanding number 2. Gametes are haploid, so contain only one allele of each gene. And understanding number 3. The 2 alleles of each gene separate into different haploid daughter nuclei during meiosis. Okay, these are basically taken right out of topic 3.3. We talked about this already. So remember, meiosis results in haploid gametes. So they have half the number of chromosomes that you would typically need, right? Half the chromosomes. Half of those chromosomes means you only have one allele for each gene. So let's think about your eye color for a minute, right? You, when you were a zygote, you were the combination of the egg from your mom and the sperm from your dad. And if you're uncomfortable with thinking about that as like your parents and you, then think about something else. Think about like your pet or something. In any case, so here's a zygote, right? 46 chromosomes. Half of those chromosomes came from the egg, half of those chromosomes came from the sperm. I have blue eyes, which means I got the gene for blue eyes from both my parents because that's a recessive allele. You can only see it if you have 2 recessive alleles. That means one recessive allele came from my mom and one recessive allele came from my dad. So you have 2 copies of each gene, right? One from your mom, one from your dad. That's basically what they're trying to say here. So remember, meiosis results in haploid gametes, which contain only half the number of chromosomes you need, and one allele for each gene. So you can think of it as this is one copy of every gene needed to make a human. This is one copy of every gene needed to make a human. This is 2 copies of every gene needed to make a human. Sometimes genes cover each other up, right? And that's the rest of the lecture today. During gamete production, the 2 alleles for a gene segregate into different daughter nuclei. The overall combination of alleles resulting in cells is random, promoting variation. And this is the second law that we talked about, right? So we talked about 2 laws a minute ago, the law of segregation and the law of independent assortment. This particular slide right here, this is the law of independent assortment. Okay? So if this is the original germ cell, we go through DNA replication. Before meiosis 1, we have meiosis 1, anaphase 1, telophase 1, right? And then we go through the whole process again, meiosis 2, prophase, metaphase, anaphase, telophase, and we end up with 4 separate gametes, right, with different combinations of genes. Understanding number 4. Fusion of gametes results in diploid, that would be 2N, zygotes with 2 alleles of each gene that may be either the same allele or different alleles. All right, so these are terms that we already learned last week. When gametes fuse, egg plus sperm, the resulting diploid zygote has 2 alleles for every gene. These alleles can be the same or they can be different. So we have 2 of the same, right? 2 of the same is homozygous. Homo means same, right? Over here, 2 of the same, homozygous. Homo means same, okay? Here we have 2 different alleles. Hetero, different. 2 different alleles. Hetero means different. So, heterozygous, the 2 alleles for a gene are different. Or homozygous, the 2 alleles for a gene are the same. Now, it can be homozygous dominant, like over here, or it can be homozygous recessive, like over here. As long as the 2 alleles are the same, that's homozygous. Okay, take a minute and see if you can do these on your own. All right, here we go. So we have capital A, homozygous, capital B, lowercase b, hetero, capital C, lowercase c, hetero, 2 uppercase, homo, uppercase, lowercase, hetero, 2 little f's, homo, big G, little g, hetero, big H, homo, big I, hetero, big J, hetero, little k, homo, big L, homo. And I know everybody was like, no homo, get over it. Okay, understanding number 5. Dominant alleles mask the effect of recessive alleles, but codominant alleles have joint effects. Okay, let's break that down into 2 separate parts. So when we're discussing inherited traits, there's 2 important terms that are used. Genotype, that's the combination of genes. Alleles are genes. Remember, alleles are versions of the genes. Genotype are the genes that you have. Your genotype, the type of genes, right? That makes sense? The genotype is the type of genes that you have. And then there's the phenotype. And the phenotype is what you can actually see with your eyeballs. Now, sometimes you can tell an organism's genotype by looking at its phenotype, but not all the time. So if it's a recessive phenotype and you can see it, like my blue eyes, then you know what my genotype is. But if I have brown eyes, that doesn't mean I have 2 dominant brown-eyed genes. I could have 1 dominant and 1 recessive. I could be heterozygous. So sometimes you can interpret genotype from phenotype, and sometimes you can't. If you know the genotype, you can always tell what the phenotype will be. Did I confuse you? So genotype is the genes that you have. Phenotype is what you actually see with your eyeballs. So, some alleles are able to mask the effects of others when they are present. These are called dominant alleles, and they're represented by capital letters. So, for our pea plants, purple is dominant, right? And lowercase a is recessive. Recessive alleles are those that are masked by dominant alleles. They're represented using lowercases. So, here is a homozygous dominant, capital A, purple flower. This would be considered, Mendel would call this a pure purple flower, right? 2 capital A's. Here's our heterozygous, 1 of each, capital A, lowercase a, heterozygous. It's still purple because this big A covers up this little a. Here we have homozygous recessive. We have 2 little a's, right? 2 little a's are going to be white. There's no capital A to cover them up. So we show the recessive phenotype, the white phenotype. Okay, So now you're going to practice writing some phenotypes. That means what you would see. Purple flowers are dominant to white flowers. Brown eyes are dominant to blue eyes. Round seeds are dominant to wrinkled seeds. And bobtails in cats are recessive, meaning a stub tail in a cat is recessive. Long tails in cats are dominant. Okay, so take a minute and see if you can answer these yourself. Okay, let's see what you got. Did you get them all right? I got them all right. Okay, so now let's tackle the second part of this understanding. We talked about dominant alleles masking the effects of recessive alleles. Now let's talk about codominant alleles. Codominance occurs when codominant alleles are present. These have joint effects and are both seen in the phenotype. In the example here, and I think I talked about this last time, we can see that red and white alleles mix to form a pink flower. So we have our phenotype, which is red, our genotype, capital R, capital R. Capital R, red is dominant, right? And we have white over here, which is lowercase r, lowercase r. The difference is when you have a heterozygous pair, instead of the big R covering up the little r, they're both expressed, and you get pink. Now, sometimes you'll get stripes or spots, and that's something different. That's not codominance, that's incomplete dominance. And we'll talk about that another time. For now, if you have codominance, you see a mixing of the two genotypes. We talked about this last week with the nose size, right? So if you have a big nose, like a big honking nose, and your spouse has like a tiny little nose, right? And your kids get one of each, they're going to have average size noses. Unless both parents are like, if both parents have normal sized noses, so then your kids are in trouble because they could have giant noses, small noses, or medium noses. So, keep that in mind. All right, so if you want some more resources and how these inheritance patterns work, there's a really good TED Talk, TED Ed on Mendel's pea plants in particular, especially if you're trying to study up for that skills question. And then there's always Crash Course. They do a good job of explaining heredity too. And you normally would be able to like click on these, but you can't because, you can't. But if you just search it on YouTube, they'll both pop up. Okay? All right. So next, let's talk about monohybrid crosses. Skill number 1, construction of Punnett grids or Punnett squares for predicting the outcomes of monohybrid crosses. We spent a lot of time doing this last week. So Punnett grids or Punnett squares are a tool that can be used to predict the outcomes, right? You can predict the percentage, the chances of the offspring of monohybrid crosses. So when we say monohybrid, what do you think that means? Mono means one, and hybrid means two different kinds. So what you're going to get is parents that are heterozygous. So monohybrid crosses mean parents that are heterozygous. Okay? And we're only going to look at one trait at a time. So our trait is flower color, our gene is capital B, and our alleles are capital B, dominant purple, and lowercase b, recessive white. Okay? And as you can see, we have 50% heterozygous, 25% homozygous dominant, and 25% homozygous recessive. Now, if we were looking at phenotypes, we would say 75% purple, right? Because there's 3, and 25% white. I think we did that exact same one last week. So, as a review, let's see if you can do one. So, actually, I'm going to show you first, even though I already showed you. All right, so for a particular trait, let's say R, we're going to determine the genotype of the parents and write them as a cross. So if we're doing monohybrid crosses, right, that means both parents are heterozygous, right? So write the genotypes of the parents on the top and left side of the Punnett grid. There should be one allele per box on each side. Here we go, 1, 2. Then you're going to use the genotypes on the outside of the grid to fill in the squares, just like that. And 4, determine the number of each type of genotype and phenotype. And we're using this to predict the ratios of the offspring. So we would expect our phenotypic ratio. Here, let me see if I can use my cool pen and draw on here. So we would expect our phenotypic ratio to be 75. Oh, it went away. 75% purple and 25% white, if this were flower color, right? Okay. Okay, now it's your turn. A heterozygous yellow pea plant is crossed with a homozygous green pea plant. You have to know what's dominant and what's recessive. Hopefully you wrote down those 7 traits in the beginning of the slideshow, and you know which one is dominant and which one is recessive. If not, you can go into OneNote and look at the slides there, because they're already there. All right, take a minute, work this out on your own. Okay, let's see what you got. So we have yellow, which is dominant, and green, which is recessive. We have homozygous green. Homozygous green. Okay, that means that we have capital Y, lowercase y. Homozygous green. That's this one. Lowercase y, lowercase y. This is the green one. Right? Heterozygous yellow. Here's our heterozygous. Homozygous green. That's this one. Okay? So heterozygous yellow. Homozygous green. Okay. Put those there. Right? Fill in the square. Okay. Let's look at our results. Genotype. Phenotype. Cool. Let's do one with eye color. So, a heterozygous brown-eyed man reproduces with a homozygous brown-eyed woman. What portion of the offspring will be blue-eyed? Oh, look at that. It's right there for you. Freebie, huh? There you go. Okay, the next one we're going to talk about is blood type. And we talked about this a little bit last week too, right? Human blood types are determined by the presence of proteins on the surface of red blood cells. The blood types of parents' offspring can be predicted using monohybrid Punnett squares. All right, so let's just back up for like a hot minute, right? So do you know your blood type? I know my blood type. I am B positive. So think all the way back to first semester when we talked about the cell and we talked about proteins on the surfaces of the cell membrane that are used for all sorts of things. There's integral proteins that span the membrane and they help push stuff in and out. We have surface proteins that are used for cell communication. This is an example of one of those surface proteins, and it's really, really important. So you have 4 different blood groups, right? Group A, Group B, Group AB, and Group O. So if you have type A blood, you have a particular kind of surface protein on your red blood cells. You also have antibodies in your plasma that are anti-B. So they are anti this guy, right? So you have this kind of protein and you have antibodies that will attach to these guys and flag your immune system to attack these guys. And that's why if you get a transfusion with the wrong blood type, you're in big trouble because it's going to cause a clot and you're going to have a stroke or a heart attack like immediately, because all these antigens right here, antibodies, they're going to grab right onto this cell. Your immune system's going to freak out and everything is going to get clogged up. So if you have type A proteins, right, you are blood group A. If you have type B proteins on your blood cell, you are blood type B. If you have both, check it out, your cells have both. right? So these guys have anti-A. If you have both A and B, can you guess what kind of antibodies you have in your system? You have none. It would not make sense for you to have either of these because you have both A and B on your blood cells and you don't want your immune system attacking your own blood cells, right? So if you have AB, right, you have no antibodies. If you have group O, that means you have no proteins on your blood cells. They're smooth like a baby's butt, right? And you have anti-A and anti-B. So you cannot receive blood from this person, or this person. In fact, the only person you can receive blood from is this person right here. They say carbohydrates, but it's really a carbohydrate protein complex. Okay, so there's 4 blood types in humans, A, B, AB, and O. The genotype for each is shown in the table below. Each allele is represented with an I with a superscript. So I, capital A, because it's dominant, right? Superscript, or lowercase I, which is lowercase. That's the recessive. So this is an example of codominant. Both A and B are dominant. So if you get both A and B together, they're going to express together. You're going to see both. One does not hide the other. However, if you have a lowercase I, those guys can be hidden. So here's a lowercase I with a capital I or a capital A. This capital A hides, right? You're adding that protein on. You're not bald anymore. You're adding that A protein. So I'm going to let this play for a minute. I just want you to take a look and see if you can absorb what this diagram is showing you, and then we'll talk about it in a minute. Okay, so when you donate blood, that blood can only go to specific people. So here we're starting with, we're already in A's. Okay, next is going to be B's, right? So if you have, and I probably should back up and say, the plus minus thing is another genetic factor. It's the Rh factor, and it follows kind of the same rules that the blood type does. So if you are positive, that means you're Rh factor positive. You can receive either positive or negative blood. If you are Rh negative, you cannot receive positive blood. So what you're seeing is if you have A minus blood, that's who can receive your blood. Here's B positive, that can go to B or AB plus. B negative can go to anybody with a B. O positive can go to anybody that's positive. O negative can go to everybody. O negative is the universal donor. AB positive can only go to AB positive. AB negative can go to AB positive or negative. A positive goes to A positive and AB positive. A negative goes to those guys. So let me see if I can summarize this. If you have type A blood and it's negative for rhesus factor, you can give it to anybody else that has type A. If you have type B negative blood, you can give it to anybody else that has any variation of type B. If you have type O negative blood, you can give it to everybody because your blood doesn't have the surface proteins that's going to trigger the immune system. Now, if you have positive blood, whether it's A, AB, or O, you can only give it to other people that are positive. People that are negative are going to have a reaction to it. Now, you may be wondering what these white numbers are off to the side. These are the proportions of the global population that have that blood type. So 39% of the population has O positive, 27% has A positive, 22% has B positive, 6% have AB positive, 3% have O negative, 2% have A negative, 0.6% have B negative, and 0.4% have AB negative. So let's think about that for a minute. This person right here, this is a universal donor, meaning blood from this person can be donated to any other individual on the planet. But look at that percentage. It's really, really low. So there's not very many people that have that. Similarly, you can kind of see that all throughout this entire site. So these collectively, these negative, Rh factor negative blood types have much smaller proportions than these Rh factor positive types over here, right? This is why it's so important to donate blood, because the blood that you're donating is so very specific. It can only go to a certain portion of the population. So if you have the ability to donate blood, please do. If you can't, then you can't. But if you can, this is what helps keep people alive, particularly in pandemics and situations like this. Okay, so let's see if you can do another example. Here we go. Punnett grids for blood types are used the same way as other monohybrid crosses. For example, this grid shows the results of crossing two AB individuals. Let's see if you can do it on your own. Okay, let's see what you got. So there's IAIB, IAIB. Let's look at our genotypes and our phenotypes. Genotypes and phenotypes. So 50% codominant blood type AB, 25% type A, 25% type B, right? So here you can see we have 50% here, 25, 25, and that matches down here, 50%, 25, 25, because this is a co-dominant relationship. Nothing's hiding anything else. Nobody's being hidden. Let's do another one. If a heterozygous type A male mates with a heterozygous type B female, what percentage of the offspring will have type O? Let's see if you can do it. You should have come up with something like that, right? All right, so if you need a couple more resources and you have some time on your hands and you're more interested about what blood types are, here are some links. You can talk about why blood types, or you can listen to why blood types matter. There is some recent research in this particular field that talks about the evolutionary advent of the type O. Type O, I think, is the most recent, and there is some indication that this may provide you somewhat of an advantage in malaria-stricken areas, similar to how sickle cell provides you an advantage. But I think that research is relatively new. Somebody once asked me, does your blood type matter in terms of infection? And I think it really depends on how the pathogen is invading, right? So if that pathogen has some sort of connecting protein that prefers certain surface proteins on your blood cells, then absolutely. But off the top of my head, I couldn't tell you one of those. That doesn't mean it doesn't happen. It just means I don't know. All right. So now let's talk about some of the diseases that occur with these mutations and how they may or may not be associated with sex chromosomes. So, understanding number 6. Many genetic diseases in humans are due to recessive alleles of autosomal genes, although some diseases are due to dominant or codominant alleles. And we're going to talk about understanding number 9 as well, which is many genetic diseases have been identified in humans, but most are pretty rare, right? So, oops, there we go. Okay, genetic diseases are disorders that are caused by errors in the genome. And we talked about that before. We talked about that when you have point mutations, substitutions or deletions, frame shift mutations, anything that's not a silent mutation could cause a genetic disease. Sickle cell was the one we talked about the most, where you have that one base pair change, right? And that changes the entire shape of the red blood cell. Most genetic diseases result from recessive alleles on autosomal chromosomes. So that should trigger a memory for you. Autosomal, that means pairs 1 through 22 chromosomes, right? Some are dominant, and we'll talk about examples of both recessive and dominant genetic diseases in just a few minutes. So here we have an example of an autosomal dominant disease. If you have a one parent that has just one copy of the allele and a mother that has none, chances are 50% of your offspring, let me rephrase that. There is a 50% chance that your offspring will obtain one copy of that dominant allele, and thus they will express the disease, right? Now let's look at autosomal recessive diseases. Here we have two parents that are carriers. You can see there's the little orange one right there, or yellow gene right there, and there's the little yellow gene right there. Each parent has one copy, right? That means there's a 25% chance that the offspring will receive 2 copies and show the disease, but there's also a 50% chance that the offspring will inherit 1 copy, whether it's from mom or from dad, right? These guys would be carriers, meaning they would carry the disease, but they would not show it because this healthy chromosome would, or this healthy gene would cover up this recessive gene. There's also a 25% chance that their children will be totally normal. So, anybody's guess. And again, remember that most genetic diseases are pretty rare in humans, and our improving technology allows us to identify these and study them more. Think back to 3.3 when we talked about chorionic villi sampling and amniocentesis to develop karyotypes, where we can actually look at chromosomal abnormalities in individuals, be it from their regular autosomal tissue or their gametes, or even in a fetus. Okay, one of the diseases we're going to talk about, this is A3, inheritance of cystic fibrosis and Huntington's disease. So cystic fibrosis is a genetic disease that results from recessive alleles on chromosome number 7. The presence of this allele produces extra chlorine ions channels, not the ions themselves, but extra channels that increase chloride levels in sweat. And it decreases levels in mucus. And what that does is it causes the mucus to thicken up. So here we have chromosome number 7. How can I tell? Because there's a whole bunch of 7s. And it's telling me P, see that P arm? The P arm is on top. The Q arm is on the bottom. This is the centromere. That's the anatomy of a chromosome. You can see these are loci. 22.2, 21.1, 14.3, 12.3. Those are locations of different chromosomes. Here is, I believe right here, 7q31.1. So that's the 7th chromosome, the Q arm, the bottom position, 31.1, right? I believe that is the location. It could be right here, but I'm pretty sure that's it. That's the location of the allele for cystic fibrosis. If you have the mutated allele, right, it's recessive. If you have just 1 copy, you're okay. If you have 2 copies, what happens is you have these extra ion channels, right? And you get this buildup of mucus. So mutant CFTR channel does not move chloride ions, causing sticky mucus to build up on the outside of the cell. So sticky mucus, right? That's not so great. This causes all sorts of problems. A normal channel moves chloride ions to the outside of the cell. It helps prevent this mucus from caking up. I think I said it backwards a minute ago. So if your chloride ion channel is not working properly, right? It's not pushing those chloride ions out, and you get a buildup of mucus on that cell. Remember, this is a membrane of a cell. So on the outside of the cell, you get that buildup of mucus. And that mucus can cause a lot of problems. So you frequently get bacterial infections that kind of fester in that mucus lining. People have trouble breathing, decreased gas exchange. We'll talk more about that in topic 6. The air sacs in your lungs, the cells that actually help exchange gas, are very, very thin, and any buildup of any kind of material on top of that cell decreases the ability of gas to exchange across the membrane. All the diffusion of oxygen into your body and carbon dioxide out of your body, that's all passive diffusion. There's no active transport there. So if anything gets in the way of that, it really decreases the amount of oxygen that gets into your system. A lot of people are having trouble with that right now. One of the things that's causing COVID-19 to kill people is that you're getting a mucus buildup in the lungs, and people are literally suffocating to death. But anyway, other issues, the pancreatic duct also gets blocked with that mucus, and that can cause trouble digesting food and abnormal pancreatic activity. So your pancreas, the job of your pancreas is to produce a bunch of enzymes that help you digest your food. And if that mucus blocks that lining, it's like a traffic jam. Those enzymes can't get out. If your pancreas bursts, those enzymes start digesting your actual own tissue. That's not good. And if those enzymes aren't getting into your small intestine where they're supposed to be, you are not properly digesting your food. You're not getting the proper nutrients out of your food that you need, and you can actually end up with all sorts of nutritional issues like scurvy and stuff, weird stuff. So here we have a normal airway. You can see this is a alveolar, or these are alveolar sacs, and here you have a bronchial tubeway. This is healthy. There's nothing impeding the airway. These little bubbles are where gas exchange actually happens. So gas comes in through the esophagus, down into the lungs, out into these little tree branches, right? And these little bundles here, that's these alveolar sacs. When a person has cystic fibrosis, when they have two copies of that allele, those chloride pumps don't work, so you're not pumping chloride ions out as much, and that causes fluid and mucus buildup, and bacteria thrive and stuff like that. So if this is not cleaned out properly and you get this buildup, you get a bacterial infection. You can also get blood in the mucus. Your immune system starts causing inflammation and swelling, similar to asthma. That's all going to prevent oxygen from getting in here and diffusing into your bloodstream. I had a friend in high school that passed away from cystic fibrosis his sophomore year. It was pretty sad. He was a nice guy. He played really, really good guitar. It was a shame. Okay, so just like with any other trait, we can use a Punnett square to help predict the probability of offspring inheriting based on the genotypes of the parents. So, since cystic fibrosis is an autosomal recessive disease, Punnett grids can be used to determine how the trait will pass from parent to offspring. Chances of inheriting the disease are not related to gender. So this is an autosomal chromosome. This is not a sex chromosome. Doesn't matter whether you're a girl or a boy. So here we have a carrier father, right? Heterozygous. And here we have a carrier mother, heterozygous. They may or may not know that they carry the gene. Since they're heterozygous, they probably don't exhibit the disease. And if nobody else in their family happens to exhibit the disease because they're also heterozygous, there's no way that they would know. This happens more often than you'd think. So here we have the Punnett square. We have the heterozygous mother, the heterozygous father. You can see the chances down here. We get unaffected, right? So this is our unaffected son right here. Here we have a carrier that's a daughter. She's got one copy. We have a son that's a carrier. And I say son and daughter, and it really doesn't matter. You could switch around the genders of these kids and it wouldn't make a difference. What the reality is that you have one child that's unaffected, 25% chance of being unaffected. You have a 50% chance of your children being carriers, and you have a 25% chance of your children having the disease. That Jeff is the name of the student that I just talked about that passed away my high school year. He had 10 brothers and sisters. And I remember when I first learned about this in high school, actually, and it was the year after that he had passed away, because he was in a different grade than I was. I was kind of shocked because he had so many brothers and sisters, and the parents had to know that this was coming from them, and yet they continue to just have a bajillion kids. I couldn't understand it. This was the late 90s. Genetic counseling was a thing. I mean, it was relatively new-ish, but they could have done it. And I just remember feeling angry because I had just learned that they had knowingly continued to have kids when they knew that they could pass this on, even though they were both fine. Okay. Next, let's talk about Huntington's disease. So Huntington's disease is a genetic disease that's caused by a dominant allele. So you just need one copy. And this allele occurs on chromosome number 4. So because the allele is dominant, any individual that has even one copy is affected. So here we have a male and a female. We have gene positive, gene negative. So let's figure out what that means. Gene positive, right, means that they have the mutated allele. Gene negative means they do not. So here we have, and this kind of looks like a pedigree. We'll talk more about that later, right? We have a male that has at least one copy, right? So we don't know though. We don't know if he's heterozygous. Let's write some stuff down and see what happens. I'm going to use my handy dandy new pen. Okay. So let's make the gene. We'll make it. Oop, nope. I gotta hit the pen thing first. Pardon my noobness with my new stuff. It's actually not even mine. It's my daughter's. I just borrowed it. Okay, here's the pen. All right, so we're going to use H to be... that's not good. That's a capital H. So that's positive, meaning they have the disease, and that's lowercase, meaning he doesn't have the disease, or he, she, it. All right, so here we have a guy, right? He could be literally, he only has to have one copy, so he could be heterozygous, or he could be homozygous because he only needs one copy. Now, here's a woman who does not have any copies, so we know that she's got to be little h, right? And they have a son. He looks like he's got at least one copy. Well, we know if mom does not have any mutated versions of the gene to give, that she can only give little h, right? So that means he's got to be heterozygous because he has the disease, right? He's light blue. He has the disease, but he can only get a little h from mom. Now he marries a woman that has no disease, right? And here are their kids. Here's one kid that definitely has one allele from her dad that's big. All right, and there's none. So you can see how this kind of works. Here we have a guy, looks like he's negative, so he's got two little h's. He marries somebody else that has two little h's. Well, if all they have are little h's to give, then it makes sense that all their kids are just little h, right? Here we have a daughter, and it looks like she got the dominant allele from her dad. Here we have another daughter that got, oh, look, we know that he has to be, no, I guess no, we don't. Okay, he has to be heterozygous because she's normal. Wait, is that right? Yeah, that is right. He has to be heterozygous, right? He can't be homozygous because if he was homozygous, then he wouldn't be able to give her a little h. So she's got two little h's, right? So that means he's got to be heterozygous. Here we have another daughter. She has the disease, right? But she's got a little one from her mom, and she marries a normal male. And here are their children. So they can have all four. So let's see. This person does not have it. This person does not have it. These two could be, they can only be heterozygous because mom only has one to give, right? Okay. All right. So that was kind of an impromptu lesson on pedigrees. We'll talk more about that next week, actually. But you can see this is a visual diagram. You only need one copy of the gene to show the disease. And that's how that works. Now, Huntington's is a particularly nasty disease. It causes degenerative changes in the brain, which typically don't start until ages 30 to 50, which means you are well old enough to have children before this disease starts to express itself. So say you waited, say that you don't start getting symptoms until 40 or 45 and you had your kids at 30. You've got a 15-year-old kid. You already made the decision to have kids. You were normal and healthy, and then all of a sudden Huntington's pops up and it's a dominant allele and you only need one copy of the gene to get the disease. There's, a certain chance that you pass it on to your offspring. So this is pretty insidious. A lot of people don't know that they have it until after they have already have children. And that's why this disease persists in human populations. Eventually, if you have the disease, it's similar to like early onset Alzheimer's. Eventually, you require nursing care, and you usually end up succumbing to infectious disease within, 5 to 10 years of diagnosis. I think survival rate might be a little bit higher, but it is a terminal condition. Here you can see the functionality in the brain. So this is what a normal brain looks like. These red spots right here, this is the front of the head, so your eyes would be here. This is the back of your head. So these red spots that you're seeing right here are right in there, right there. And we'll talk about brain structure next year a little bit. But you can see normal activity, and you can start to see diminished activity as well. And you can actually see shrinkage as well. So the entire mass of the brain starts to shrink. You see decreased activities, larger intercranial fluid-filled spaces. It's not a nice disease. Okay, understanding number 7. Some genetic diseases are sex-linked, meaning they do occur on the sex chromosomes, the 23rd pair. And understanding number 8, the pattern of inheritance is different with sex-linked genes due to their location on sex chromosomes. So, other genetic diseases can be linked to sex, which means they are associated with the sex chromosomes, the 23rd pair. As a result, the gender, the sex of the offspring can determine the likelihood of actually inheriting that disease. And the two most common examples are red-green color blindness and hemophilia. So, red-green color blindness is a sex-linked condition that's caused by a recessive allele on the X chromosome. And since males only have one X, they're more likely to be affected than women who have two alleles for X. And I realize that might seem backwards. Like it should, like intuitively, you think, well, they only have one X, so like shouldn't they get it less of the time because they only have one chance to get it? Well, the problem is this is a recessive allele, and if you have two X's, one of those has the opportunity to cover up the other. But if you only have one, there's nothing to cover it up. So all you need is one copy of the recessive allele to show the disease, even though it's recessive. There's no opportunity for that Y to cover it up. So I'm going to flip back for a second. Here's your Y chromosome. I'm sorry, here's your X chromosome, and here's your Y chromosome. If you have say you have color blindness and it's, up here somewhere, we'll make a gene right here, right? If you have two X's, and here's the gene for color blindness, right, and it's recessive, this X is going to cover it up. So you're not going to see it. If this is the recessive allele for color blindness, right, you have one copy, but this little tiny Y is not going to cover that up. So this is going to express. There's nothing here to cover it up. So if you're a male and it's an X-linked chromosome or it's an X-linked gene, you only need one copy because there's nothing to cover it up. Okay. So here we have an unaffected male, right? So he's got no color blindness issues. And we have a mother that's a carrier. So it looks like that's the carrier. Yep, right here. So this is the color blindness gene right here. All right. So, but she's not colorblind because this one covers that up. Okay. They have children. Should I just draw the Punnett square or can you see it? Can you kind of see it? I think you can kind of see it. And it's also on your Mendel diagram from last time. So here we have a son, and it looks like he got dad's Y. That's why he's a son, right? And here's the X, but he got mom's healthy X. Here's a daughter. She got dad's healthy X and mom's healthy X. Here's a daughter that's a carrier. She got dad's healthy X right here, and she got mom's affected X. Here's the gene for colorblindness, but she is not colorblind. Why? Because this is going to cover that up. She's just like her mother. And here we have a son who got the affected X from his mom, and he got a Y from his dad, but that Y does not cover that up. So he is actually going to exhibit signs of colorblindness. And in case you were wondering, sorry for those of you that are colorblind. I don't mean to be like a jerk about it. For those of you that are not colorblind, this gives you an idea of what red-green color blindness actually looks like. So this is what normal vision looks like: green, yellow, and red. And if you're colorblind, you can see what it looks like down here. If the top row and the bottom row looks the same to you, then you are probably colorblind, and you can give me a call and we can tell you what to do about that. We'll actually learn next topic about some new therapies, some new genetic therapies that can help reverse some forms of blindness. And I know that there are glasses now that can show you color. But yeah, so if these rows look the same, hate to break it to you, you might be colorblind. All right, so those with the disease have mutated genes that cause malformed proteins, and those proteins are used in the parts of the cells that actually receive color. So the parts of the cells in your eyeball, on the back of your eyeball, that interpret visual signals as colors. So when they send that information to the brain, they're not sending red and they're not sending green. They cannot distinguish the difference between those. Okay, here's an example that you can do on your own. If a male with color blindness mates with a carrier female, what percentation? Is that a word? Percentation? What percentage of their sons will have the trait? Go ahead and do this on your own. Okay, let's see what you got. You should have first written down the genotypes of the parents, right? And put it on your Punnett square. Filled it out. And that's what it looks like. So, hurrah for those of you that got it right. Now we're moving on to, oh, did I say it before? A2, red-green color blindness and hemophilia are examples of sex-linked inheritance. Yeah, okay. So hemophilia is another genetic disease that's caused by a recessive allele on the X chromosome. And this disease prevents the ability to form factor VIII, which is an essential catalyst for blood clotting. So here you have a normal blood vessel, not like a blood cell, but like a long tube, and like you cut it like you're looking down a straw, right? That's a normal blood vessel. If you get a hemorrhage, meaning the wall of that blood vessel bursts, like an aneurysm, that's a good example, your blood clots that hole so you don't, bleed out. If you have hemophilia, sometimes you'll see it spelled this way, sometimes you'll see it spelled that way. It's the same thing. Here's that normal blood vessel. It hemorrhages, right? And then there's no clot, so you could actually bleed out. Now, a hemorrhage doesn't have to be like a huge thing, like you think of like somebody having like a brain hemorrhage or like a coronary hemorrhage or an arterial hemorrhage, and like they bleed out in like a second and they die. That's not always how it goes. You can have a small hemorrhage, just a small little leak, but you can still bleed out. So this is like, you break a bone, you break an internal bone, or you break a rib, and it, nicks a blood vessel. The sharp edge of the bone nicks a blood vessel, and you don't even necessarily know. And a couple of days later, you drop dead because you've bled out internally because this little hemorrhage right here never healed. Now, this is a affliction that dominates many of the royal families, because who do royal people marry? They marry other royal people, so their family trees and pedigrees are kind of twisted, and there's lots of like weird diagonal lines, and so that recessive allele gets concentrated, and you have lots of particularly male lines to the throne, which have been kind of messed up because of this particular disease. So I'll leave that to history class for you guys. So individuals with hemophilia have a life expectancy of about 10 years if they're left untreated. So you can imagine if you are the king of a European nation and you're trying to have a whole bunch of sons and they're not living past 10, your legacy is in danger. Now, in modern day, we do have treatment for this particular disease. We can give you an infusion of factor VIII that's isolated from donor blood sources. Now, like I mentioned earlier, those donor blood sources have to be of the same blood type as the individual. So one more time, I'll say it again. If you have the ability to donate blood, please do, because lots of people's lives depend on it, not just like after a car accident. Some people need repeated therapy throughout the course of their life. Okay, here's another example. If a female with hemophilia mates with a normal male, what percentage of their daughters will have the trait? So H is normal, lowercase h is hemophilia, meaning it's a recessive disorder. It's sex-linked, right? You have to figure out, or look back a slide or two and figure out what chromosome it's on, and give it a shot. Okay, sorry about that. I was choking on my ginger ale. All right, so here are our parents. Stick them in the Punnett square. You can see we have a mother, right? Female with hemophilia. She's got 2 copies of the disease. She's recessive. I'm assuming in modern day, otherwise she wouldn't be really old enough to have kids. And she mates with a normal male, so he's normal. What is the percentage of their daughters that will have the trait? So here we go. So here we have daughter 1, daughter 2, right? These are sons over here because they have Ys. So sons, 100% hemophiliac. Sorry, guys. Oop. Daughters, 0% hemophiliac. Why? Because they got one of those H's from their, one of those X's with an H on it from their dad. They got the healthy gene from their dad. Example number 3. If a female, I really got to spell check this stuff. If a female carrier for hemophilia mates with an affected male, what percentage of their daughters will have the trait? All right, there we go. All right, hopefully you got that right. Okay, this is where we're going to stop for today. This is a slide that kind of previews what we're going to talk about next week. Next week, we're going to spend a whole lot of time on pedigrees, and you're going to learn all about the blue people of Kentucky. And I don't mean just like kind of blue, I mean really blue. Like, this is not his hand, this is somebody else's hand, this is this person's skin. You can see this guy, he's not sick, he's just old. That's why he's like little and skinny. He's just like super old. And you can see here, these are people that are actually blue, like not makeup, like they're actually blue. There's a genetic cause behind that. We're going to do pedigrees next week and figure out what's up with these blue people from Kentucky. All right. So, as always, if you have any feedback, that would be great. If you have any questions, you can type them into this comment box, and then I will look at them, and then we can talk about it on Zoom, or you can talk about it via Etsby message. Please make sure you get a hold of me this week. And as always, I miss your faces and I miss your voices. I hope to see you soon, and stay healthy.